Well, buongiorno, and I thought this morning what I'd do is I'd shift gears completely and tell you a story, just to kind of change the direction a little bit. And in deference to my two previous speakers, I wanted to tell you something about what happened in London a couple hundred years ago. There was a chap by the name of John Hunter, and he was a very famous pathologist. And so John Hunter, there's a Hunterian Museum in London. If you go to the Royal Society, the College of Surgeons, there's an amazing museum there with skeletons of different species. And what John Hunter did that was so remarkable was the way he thought, and he looked for exceptions. So there was a giant in a local circus, Anton the Giant, and what John Hunter did was to follow Anton throughout Europe, waiting for him to die. So Anton would be there in the circus, look out the window, and there'd be John Hunter, the light on, waiting for him to die. And the reason that John Hunter did that was because this Anton the Giant was an exception. And he thought by understanding this Anton, he'd be able to understand a lot about disease, and Anton was a key to a door to understand some fundamental topics. So Anton ultimately died, and John Hunter cut him apart and found this pituitary tumour, and thereby opened this door to understanding hormonal regulation of growth. And so what we've seen the last 48 hours here are a lot of studies that have tried to understand exceptions, osteogenesis imperfecta, various diseases where there are exceptions. And today's talk is about that. It's about an exception. And we hope it'll shed some light on a fundamental understanding of a very important issue that all of us are actually wittingly or unwittingly involved with, which is that we've heard about stem cells and their value for regenerative medicine, but at the same time there are cancer stem cells. Now, when I'm a clinician basically by training, trained in medical oncology, bone marrow transplants, and we were taught that there are solid cancers and there are liquid cancers, leukemias and breast cancer, for example. But now we know that there are solid cancers with liquid phase, and whether they're gliomas, where you're looking at the local invasiveness, those patients have circulating tumour cells. The technologies that we're developing now have shown that whether you have lung cancer, breast or prostate cancer, these patients have circulating tumour cells. And that is the final frontier of the disease. Understanding these cancer stem cells is fundamental because they give rise to therapy resistance, metastasis and death. So today's presentation that I'm going to give you is based on understanding, my Anton the Giant is a fruit fly mutant. And this mutant fly is a very paradoxical fly. So I'll tell you all about this now. Now, the last 20 years, what we've been doing in my laboratory is to dissect the molecular genetic mechanisms of cancer stem cells using mouse models. And we've come up against a new mouse model which in many ways sheds lights on this fundamental difference between normal stem cells and cancer stem cells. I hope you like that story. I have a number of support mechanisms for my laboratory. So this is the way I'm going to present today, give you a brief background on the understanding of progenitor cells in the mammary gland, some of the characteristics of breast cancer stem cells, mechanisms of signaling, and some of the knockout mice models that we've developed over the last couple of decades. So this is a schematic representation of, excellent. So this is a schematic representation of the current understanding of the hierarchy of mammary epithelial cell, progenitor cell differentiation. So that there is a primordial stem cell, and it has these epitope markers on the surface of these cells, and it gives rise to these differentiated cells, ductal cells, alveolar cells, and myepithelial cells. And there are specific genes that have been identified as important regulators of these transitions using various knockout models. Now, the reason why breast cancer stem cells and cancer stem cells in general are important is that they give rise to therapy resistance and death. So understanding their properties is very important if we're going to improve the quality of life and extend life of our patients. So these cancer stem cells have these properties. They give rise to multiple cell types, they give rise to therapy resistance, they are invasive and metastatic, and they have certain gene expression characteristics which allow us to understand them. Now, if you look at breast cancer in the molecular genetic classifications we use now, we think of breast cancer now rather than by light microscopy, the histopathological changes, we look now at the genetic subtypes, and we classify them as normal-like, basal, luminal A, luminal B, HER2 positive, and the tumour subtypes in humans correspond to these precursor cell types in this hierarchy of epithelial cell differentiation. Now, my own laboratory is particularly interested in triple-negative breast cancer because we don't have any decent ways of treating that. Now, as I mentioned to you, my laboratory has been involved in defining the molecular genetic mechanisms that determine cancer stem cell, breast cancer stem cell differentiation. This is the schematic representation of the NF-kappa B pathway, and my laboratory showed that NF-kappa B is activated in a breast epithelial cell. It was known it was involved in inflammatory cells, but we showed this pathway was activated. So this is the NF-kappa B pathway, the I-kappa B kinases, the components of NF-kappa B which activate gene transcription in the nucleus. This is a theoretical model of the way in which the structure of these two proteins. So the specific question my laboratory was addressing was if you inactivate NF-kappa B specifically in the mammary gland, in the epithelial cell, not the lymphoid compartment, in the epithelial cell, what does it do to the onset and progression of breast cancer? This would be a way of telling us whether the epithelial cell NF-kappa B was important in breast cancer. And Toshioki Sakamaki made these mice. They co-integrate 5 transgenes. I think it's a world record for transgene integration. And what that allowed him to do was to turn off NF-kappa B in the mature animal. And it was important to do it this way because if you knock out NF-kappa B, you don't get a mouse. So you have to allow the mouse to develop normally, then inactivate NF-kappa B specifically in the mammary gland. So what we see here is the rate of onset of tumorigenesis in the normal mice, but if you inactivated NF-kappa B, it delayed the initiation of tumorigenesis. So this tells us that there's a very specific, distinct function for NF-kappa B in the epithelial cell, which is the initiation, not just the growth of a tumour. And it's thought that cancer stem cells are important in the initiation. It is the number of the new tumours that are forming and in the initiation of tumorigenesis, which is dependent upon these cancer stem cells. So from a therapeutic point of view, it was important to confirm this. These are assays of stem cells, and the key point here is if you look at these are a highly metastatic human breast cancer cell, and these are the number of stem cells by FACS analysis, and you can see that if you inactivate NF-kappa B, this is I-kappa B superrepressor, there's a dramatic decline in the number of breast cancer stem cells in this metastatic breast cancer cell line. And again, these are drugs which are used to inactivate NF-kappa B, and they decrease the number of cancer stem cells in tissue culture. So these were the studies that led to an understanding that NF-kappa B is turned on in the breast epithelial cell, not just the immune cells, and that it's very important that NF-kappa B is very important in the cancer stem cell expansion and the initiation of breast cancer formation. Now, we went on to define the mechanisms, and the key point is that SOX and NANOG, you've heard about SOX-OC, NANOG, c-MYC, KLF4 yesterday. So these are the 2 important transcription factor targets involved in stem cell function, and they are direct transcriptional targets of NF-kappa B. So the next target gene we defined as involved in epithelial cell cancer was the first proto-oncogene to be defined. I think Jun means firstborn, so C-Jun, people are familiar with this term, it's part of the AP1 complex, because if you delete c-Jun, you get tumours in a variety of, if you overexpress c-Jun, you get tumours in a variety of different cell types. We looked at whether c-Jun played a role in cancer stem cell formation. So this is a schematic representation of pathways, cytokines and growth factors that activate kinase signaling pathways to induce the c-Jun and FOS proteins to induce gene expression. Now, our contribution to the field was to show that c-Jun regulates not just gene expression but also gene splicing and cancer stem cell formation. So this is a schematic representation of the way in which we selectively inactivated the c-Jun gene. And these are migration assays. When we knock out the c-Jun gene, it affects the migration of cells and it reduces cellular velocity. So these are velocity studies. This is the migration of a normal cell. These are epithelial cells. If you knock out the c-Jun, there's a reduction in cellular velocity. And these are mammary stem cells, so one can assay for a cancer stem cell by the number of spheres that grow, mammospheres, or in the case of brain cells, it's neurospheres. And so the number of these and the size of these are enumerated, and these represent a quantitative surrogate assay for cancer stem cell formation. If we knock out c-Jun in a mammary epithelial cell, we reduce the number and size of these mammospheres. And if we use surrogate markers of cancer stem cell formation, epithelial cell formation, the number is declined when we delete c-Jun. Now, you heard today about the importance of secreted factors in normal stem cell function. Those secreted factors are also very important in cancer stem cell function. And these are examples of studies that we conducted to analyse those secreted factors. We demonstrated that if you take the supernatant, it's capable of recapitulating the phenotype that you see from genetic deletion of the target gene. We quantified and analysed by proteomics the proteins in the supernatant. And this is just an example of how one would do this. These are cytokine arrays, and you can see that the abundance of these particular proteins is decreased upon genetic deletion. So you can imagine doing similar things on the serum of patients to analyse changes in the specific cytokines that are secreted in the bloodstream. The key point here I wanted to make is that there's a target gene, CCL5, which is dramatically regulated by c-Jun. And if we add back this protein, SCF, it's able to restore the migratory properties of cells that are lost upon genetic deletion. So these were the studies, and the conclusion that was drawn from this is that this proto-oncogene c-Jun promotes stem cell expansion, and it does so through inducing the expression of, not surprisingly, stem cell factor, SCF1, and also regulates migration through the induction of CCL5. So the first proto-oncogene to be discovered, c-Jun, induces cancer stem cells. It does so through a secreted factor, SCF, and that thereby induces cancer stem cell expansion. The third target gene I want to talk to you about is P21. P21 CYP1-WOF1 is a cyclin-dependent kinase inhibitor. And this is a schematic representation of the way in which P21 is thought to work. It acts as an inhibitor of kinases, cyclin E and cyclin D kinase activity. Now, we conducted experiments to analyse the importance of P21 as a tumour suppressor a decade or so ago. We took mice that were deleted of P21. We crossed them to mice that had mammary tumours induced by either RAS or MYC. And we showed that deletion of P21 reduces the rate of onset by these two oncogenes, but they did not have the predicted effect on the kinase activities, the cyclin D and cyclin E kinase activity. So the point I make here is that if P21 is an inhibitor of a kinase activity, you'd see a difference, a dramatic increase, and that's what you see in the knockouts. But the opposite, so you see the dramatic increase in the context of RAS. So you delete P21 and the kinase activity increases. But in the case of MYC, you did not see that. So there was P21 is functioning as a tumour suppressor for MYC, but it's not working through inhibiting kinase activity. So that was a dogma. It was thought that the way in which tumour suppressors worked is through inhibiting these cyclin-dependent kinases, and clearly that's not the way it works in this particular situation. So what we found upon further analysis of the tumours, and I'll summarise this by just pointing at this one part of the slide, and that is this part here. If you look at the MYC or RAS tumours, P21 was inhibiting here migration. Here P21 is inhibiting migration. And we found that there was a substantial change in that P21 was inhibiting both RAS and MYC-induced stem cell number, function, and gene expression. But the particular signalling pathway to stem cells was distinct for P21 inhibiting RAS or P21 inhibiting MYC. So this is the gene expression profile here. In the case of RAS, these were the target genes that were directly regulated by P21 in the context of RAS, and these were the genes that were selectively regulated by P21 in the context of MYC. Now, the individual gene names are not particularly important, but it is important to understand that the mechanism by which P21 functions as a tumour suppressor in the context of MYC is via restraining MYC-induced cancer stem cells as opposed to inhibiting kinase activity. So this is a summary of the current understanding of the way in which P21 tumour suppressor functions to inhibit RAS and MYC-induced tumorigenesis. And again, the important difference here is that it's through a restraint of MYC-induced cancer stem cells. So now I'd like to turn to my Anton, the giant, and why this particular gene pathway is so interesting. So does anyone know what a dachshund is? Normally I ask students, but I don't think there are any students here. But so a dachshund is a little dog. It's got these short limbs, and Drosophila people are able to give interesting names to genes they discover. And so the fruit fly that has a mutant of DACH, the flies have short limbs, they have small brains, they have atretic genetics, so the ovaries are atretic, and there's generalized mesenchymal organismal development has failed in these flies. So when this gene was initially cloned, it was cloned as a dominant negative mutant of a growth factor receptor. So you can see why this is my Anton, yeah? So organismal development, all the stem cells in this fly that give rise to the eye, to the brain, to the organs are defective, and the gene is acting as a dominant inhibitor of growth factor receptor signaling. So what does every patient that we treat die of? Hyperactive growth factor receptor signaling. So this is like, I don't know, the holy grail, if you will. This gene is such a cool gene to study. And what did they do with this gene? They threw it in the trash can. No one studied it because it didn't make sense, yeah? So we've been studying this gene. And what we found is that the abundance and expression of this gene, DACH, it's expressed in humans. It's lost in the vast majority of human cancers. When we reintroduce it into cancer into cancer cells, it blocks the growth of those cells. And its activity is regulated by phosphatase, and phosphatases are very tractable from a drug therapy point of view. So this is a completely new pathway. It's not in the textbooks. It's a very new pathway, and it's known as the cell fate determination pathway, and it seems to be very important in cancer and very important in cancer stem cells. So it was cloned as an inhibitor of epidermal growth factor receptor. It's known as ellipsin in the fly. We have shown that its abundance in a variety of malignancies, just some of them are shown here. It inhibits growth. It's lost and mutated in a number of brain malignancies. It's very important in neurosphere formation. And we've shown that it restrains cancer stem cell expansion in a variety of different cancers. It binds stem cell targets. It's mutated. Now there are a number of whole genome sequencing projects. It's been found to be one of the few new genes that are frequently mutated in human cancer. Non-small cell lung cancer is one example. And we've characterised its shape, structure, its post-translational modification. So I'd just like to share with you one or two slides about this new pathway as it relates to normal and cancer stem cells. So this is I'd like to introduce you to the fruit fly eye. So this is a fruit fly eye. This is its ommatidia. And if you look very closely, high level, this is what the fruit fly looks like normally. But if you take this Drosophila gene and you put it into a precursor cell in the fly eye, it will form a new eye. And the ommatidia look very much like the natural ommatidia. So there's sufficient information. So here's the normal fly eye, and here's a fly eye developed just by putting DATCH into a precursor cell. So it's able to coordinate organismal development. It's not a perfect eye, but it's able to coordinate the abundance of all the genes. It's a master regulator of eye development. So this is the way, these are some of the interacting proteins in the pathway. So here's DATCH, and here are some of the other proteins that interact, involved in eye development, muscle, and we've subsequently shown it's true also in cancer. So this is important because it's a phosphatase and it's a tractable target, and it governs the activity of DATCH. So if you show patients with a reduction in DATCH expression, there's about a 40-month difference in survival amongst patients with human breast cancer. DATCH is a master regulator of cytokines and chemokines that you've heard about in the last day and a half that are important in stem cell function. It's also a very important regulator of metastasis. So these are the lungs of mice that have been injected with India ink post-mortem with the intention of showing the presence of metastasis. These white dots are metastasis on the surface of the lungs. So these are the number of metastases in these mice. If we induce the expression of DATCH, we block the metastases to the lungs. We looked at the mechanisms by which DATCH blocks metastasis, and it's through restraining the secretion and expression of IL-8, which is KC in the mouse. So DATCH expression is lost in human breast cancer, and DATCH1 restrains metastasis of breast cancer to the lungs of mice. And it does so through restraining the abundance of a secreted cytokine IL-8. So the question was, that we really wanted to address was what happens in the normal mouse, in the normal mammary gland, so we can understand the mammary gland gene expression and mammary gland stem cell function. So if you knock out the DATCH gene, the mice die. If you want to follow a gene in a whole animal and it contributes to new stem cells, and you knock it out, then there won't be any cells to follow. So the way in which you can deduce the function of a gene that plays a key role in normal organismal development is to mark the cells with a fluorescent protein. So to do this, we made mice that were inducible by the presence of tamoxifen, and in which the mice were normally red, but upon deletion of the gene they became green. So you can see that when you normally the cells are all red, but so this is the mouse. When you knock out the gene, the mouse goes from red to green, and so you can follow the individual cells. Normally, when you knock out the gene, there should be, so you've knocked out the red gene, and you can see all the cells in the mammary gland are now green. There's only one out of 1,000 cells that remain red. So if the gene that you knock out is very important in survival, and it promotes survival when you knock it out, you'll get a dramatic increase in the number of red cells. And that's exactly what you see here. So this is a way of tracing the function of cells which are important in survival or not. So these are types of assays that we've been doing to follow the contribution of this gene product to its progenitors. And these are types of systems we're going to have to use to be able to follow other genes that we think are important in cancer stem cells and normal stem cell function. This is shown again schematically here. So here's your mammary gland in a mouse. This is the duct. When you knock out, in this case DACH, it becomes green because you've knocked out this red gene commensurate with knocking out DACH. So let me summarize the data. I hope I haven't confused you with this color switch. But the key point here is that when we looked at the DACH floxed mice, there was a change in the structure of the gland of the mice. And the changes in the gland of the mice were the opposite to the changes in the gland of the mice of the TGF-beta knockout mouse. So we proposed that DACH in the mammary gland was restraining TGF-beta signaling. So to confirm that, we used a protein complex which is downstream of TGF-beta, SMAD phosphorylation, and we looked at phospho-SMAD activity in the DACH knockout mouse. So the increased brown here reflects the increased SMAD activity when we delete the DACH gene in the mammary gland. We did gene expression analysis, and I'll just summarize this to say that when we overexpress DACH, there are a bunch of genes that go up and a bunch of genes that go down, but the majority of these genes are genes that are regulated by TGF-beta signaling. And we went on to show that the expression of DACH, this is a TGF-beta responsive reporter, and we were able to show that DACH is capable of restraining TGF-beta activity, and that this restraint required a particular domain of the DACH protein. It does so through binding a complex. It binds SMAD4 and it recruits NCOR. So here you can see DACH immunoprecipitation and the physical association with SMAD4, which is part of this TGF-beta signaling complex. So the next question was, what about DACH in stem cells in the breast? The expectation would be that because TGF-beta knockouts show a decrease in these particular stem cell markers, that there may be an increase in the DACH mammary gland. And I'll summarize this data to say that we've looked at the stem cells in the mice and shown a change in gene expression profiling, and I've summarized this here. So in the DACH knockout mouse, these are the genes that go down. So there's a coordinated effect on the gene expression profile in the mammary gland of these mice, consistent with restraint of TGF-beta signaling. And I'll summarize this by saying that we have shown, consistent with that, that endogenous DACH inhibits mammary stem cell features. These are examples of OXOX target genes, and they are all restrained by DACH. And these are individual gene expression profiles in which those target genes you've heard about before, KLF4, Noctins, and SOX2, are restrained by DACH. So to summarize the study on the tissue-specific inducible knockout DACH mice, we've shown that there is evidence for DACH normally restraining TGF-beta signaling. It does so through inhibiting gene expression by binding a SMAD NCOR complex, which restrains key target genes involved in stem cell expansion, and thereby resulting in, upon gene deletion, an increase in the number of these mammospheres, which are breast cancer stem cell markers. So to summarize the studies we have to date on TGF-beta signaling, we have shown that this is the way TGF-beta signals, and DATCH associates with SMAD4 to restrain TGF-beta signaling and thereby restraining the branching and proliferation in the mammary gland. So this is a summary of what we know about this Anton the giant. So DATCH, this new signaling molecule, this new pathway, plays an important role in restraining TGF-beta in the mammary gland and in other cell types. It's capable of restraining EMT. It binds to displace forkhead protein binding in a variety of different gene expression studies, and it's capable of blocking lung metastasis through restraining cytokine secretion. So again, in this presentation, I've shared with you the molecular genetic analyses we've conducted to demonstrate in vivo the importance of these particular proteins as tumour suppressors that function through restraining cancer stem cell expansion. I showed you that in stem cell assays, DATCH and P21 inhibit stem cell expansion. Conversely, c-Jun and NF-kappa B in the epithelial cell expand cancer stem cells. So this is a summary of the presentation today, that at the heart of the cancer stem cell plasticity, we find the DATCH is a repressor of cancer stem cells, as is P21. So these two genes are tumour suppressors, but they work through, primarily in the case of oncogene-specific manner, restraint of cancer stem cell expansion. And two transcription factors, the first proto-oncogene and NF-kappa B in the epithelial cell, expand cancer stem cells, promoting the initiation of tumorigenesis. So with that, I'll stop. Thank you for your time and answer any questions if they're allowed. Thank you.